Okay, again, welcome to everybody. Thank you for participating this afternoon or morning, wherever you would be situated. My name is Georg Dorfner. I'm the CEO of the Siesta Group, and together with Peter Ander, our Chief Scientific Officer, we have prepared this presentation here for you today entitled What EEG Can Do for Your Trial. Trial being synonymous here almost with a pharmaceutical clinical trial, but of course other types of research and clinical trials can benefit similarly through what we are talking about today. I assume that everybody participating knows what electroencephalography or EEG is, but just to make sure that we're talking about the same thing, let me just quickly repeat that we're referring here to electrophysiological measurements on the scalp that measure brain activity, typically using electrodes attached according to the International 1020 system. A typical multi-electrode setting here would include at least 19 such leads, plus references and ground. So we would pick up multitudes of electrophysiological signals to pick up on brain activity. Some of the paradigms of measuring brain activity are done in connection with certain paradigms of presenting repeated stimuli. This is the paradigms of event-related potential. So typically one presents visual or acoustic stimuli in certain orders, for instance, an oddball paradigm in the acoustic scene or checker pattern visual stimuli to elicit responses that are called event-related potentials. That leads us to the core paradigms we're talking about here. So we can, based on whether we take single or multiple leads of EEG measurements or whether we are looking at event-related potentials in connection with a series of stimuli, we typically look at either a single channel, looking at the spectrum, perform a spectral analysis based on typically a Fourier transform. This is also called the quantitative EEG. Whereas the spectrum is then, in the next step, typically divided into bands like alpha or delta in order to get the main characteristics of the spectral properties. One cannot only do this with single channels. Remember, we have typically at least 19 such leads or more. So the next step would be to depict a topography, which is a visualization of the activity or the spectral content of the leads as distributed over the scalp. And last but not least, it can also kind of brain imaging based on a three-dimensional reconstruction of activity by brain area can be achieved using a technique, for instance, called LORETA, which would then lead us to an EEG-based tomography. And we can do this, again, as I said, with the plain resting state EEG, or we can do it by looking at the event-related potential at the main components. So this brings us to the main endpoint variables that are typically calculated here, looking at the spectrum of a single EEG channel. We can calculate the power per either frequency bin or more commonly per frequency band, like delta or the slow frequencies, alpha the more faster ones, or beta the very fast frequencies. We can calculate them in absolute or relative term, relative with respect to the power of the entire spectrum. We can calculate dominant frequencies in each band, and we can calculate certain ratios like alpha divided by theta. So these are the typical endpoints for quantitative EEG. When looking at event-related potentials, see the endpoints that are calculated are ones after averaging over several trials, time-locked by the stimuli, and we're looking at components such as N100 or N1, which is a negativity after roughly 100 milliseconds, P2, or P3 or P300, which would be a positivity after approximately 300 milliseconds. So after having had a quick look at what the main paradigms, the main measurements, and the main endpoints in EEG are, I would now like to raise the main question and at the same time, of course, answer it. Why would people consider performing EEG in a clinical trial, in particular in a drug trial? Above all, the answer would be because it's an objective measurement about drug action and drug activity in the brain. It helps you answer a variety of questions, just as, is my drug active in the brain at all? If yes, how does it act? Where does it act? At what point in time? So when? And what is my best dose with respect to an expected action? And it measures all this and answers those questions in a very cost-efficient manner. So let me have a closer look at each of these important questions and answers. The how. Here's an example, the most frequently used or seen example of how you can distinguish the type of action of a drug. If you see an increase in alpha with a decrease in delta, that is typically a sign that your drug is acting as a stimulant in some way. Whereas if you have a decrease in alpha and increase in delta, this is a strong sign that your drug is acting as a sedative. In fact, the observation that different types of changes in the EEG are correlated with different types of behavioral changes is already a very old one. So old that it's actually very surprising that EEG is not looked at like this more often than it actually is at the moment. So this is an extraction of Fink in the year 1964 already observed that certain types of increases or decreases in part of the spectrum do correlate or do correspond to certain behavioral changes that can be observed and thus indications about how a drug is really acting in the brain, what it achieves, what it does. The how can also be a related question with respect to event-related potentials. Here we can answer a question like, is a drug enhancing perception and or cognition? So we know, for instance, that shortening certain latency points toward P300, for instance, points toward cognitive improvement or perceptual improvement. So again, a very important information that you would be able to read out of your EEG paradigm. The question of where can best be answered if we look at the distribution of activity in what I already mentioned as topographic depictions. What you see here are several circular images which schematically depict the scalp viewed from above. The top here would depict the nose, and this would be the Indian in the back. And the color code is an interpolation between the typical 19 electrode positions with respect to a certain variable or even a significance value when you look at differences between conditions you would like to depict. So in this case, for instance, it would be significant changes, either positive if it's red and purple, or negative if it's blue. And you can see that certain changes, depending on whether you're looking at delta power, theta, alpha, or beta, you would see that these are expressed most strongly in different parts along the scalp. And that gives you an indication of where the activity is most strongly expressed. I should mention that in this, the main reason in this depiction for looking at different positions is to be sure to pick up the strongest activity, because ad hoc, usually a priori, one can never be sure on which of the many electrode positions an effect would be strongest. So this is actually one of the main reasons for looking at multiple electrodes and not just one, because you cannot easily know where you would see the strongest effect. If, however, you really want to be interested in brain positions, then you would probably have to look at the EEG in the tomographic aspect. And the technique that I mentioned already, LORETA for low-resolution electrotomographic analysis, would be able to depict the actual brain area in a 3D depiction of the brain, like here, for instance, the top view, the side view, and the front view, and you would obtain, and you can obtain, a kind of imaging facility based on the EEG channels. So even that is possible based on electrophysiological measures. Let's talk about the when. This is the next, another important question. Not only is EEG by itself has a very high temporal resolution, it also is a procedure that doesn't take all too long, so it's very easy to repeat it at several time points during the day. So here's an example of where you would do EEG measurements like 15, 30, 45, and 60 minutes, and then 1.5, 2, 2.5, 3 hours, and so on after drug dosing. And each of the time points, if you represent your activity as a summary topographic map, you can clearly see at what points in time the action reaches the peak. And so you would, in the end, get an interesting PK-like profile, but with the difference to the typical pharmacokinetics is that you're not measuring concentration in the blood here, but you're really measuring the activity of the drug in a temporal manner. So this is why authors here typically talk about PK/PD modeling, another interesting feature of EEG measurements. Dose finding is another important aspect of where EEG can support you a lot, where again here as an example, you have topographic maps over different frequency bands at different doses given to a subject. And again, you can see that, for instance, here the third level of dose would reach the maximum amount of activity, the maximum amount of effect, and would be able to help you finding optimal doses. This is an example of a publication by Saletu et al. 1994, using alprazolam as an example, but of course all CNS-active drugs would show similar profiles here. I mentioned that you can do all this, answer all those interesting questions in a very concise manner, but do it in a very cost-effective way. If you look at what are the other objective measures to measure brain activity, they are mostly the typical other brain imaging methods like PET scans, functional MRI, or magnetoelectrograms, encephalograms, MEGs. Compared to all these, while at the same time being a direct measurement, you are portable, and above all, the costs are the lowest, and the availability is therefore very high. So you really have a competitive instrument here in the form of the EEG that compete in many respects with brain imaging at a much lower cost and at a much higher portability. Other advantages for EEG include that the measurement is more or less largely unobtrusive. It does not bother the subjects all too much. It doesn't harm them. There's no radiation or other additional exposure to drugs. So ethically, usually the use of EEG in a clinical trial is practically without problems. I already mentioned it, that the measurements don't take a very long time. At the longest typically would be like 8 minutes per paradigm. So you can make repeated measurements without too many burdens. And the effects that I also have shown you are typically visible already with sample sizes and of like between 10 and 40, your typical early phase cohort or group size. So the method lends itself very nicely for early phase measurements of all those effects. Let me, before I come back to those aspects, takes the whole EEG paradigm even a step further beyond the questions that I've already shown you that EEG can answer, and introduce you a principle that was coined by one of our shareholders, Professor Saletu here in Vienna, the key lock principle, that he has found a lot of evidence that representative drugs of the main psychopharmacological classes induce very typical patterns in terms of changes in the EEG on one hand, and then on the other hand, the patterns of how different mental neurological disorders change brain activity in the EEG, that from these observations, the two very often complement each other and give you a very good indication about which disease or pathological picture a certain drug or drug class might be best targeted at. Here's an example just to give you the impression here. Again, there's columns of different topographic distributions divided again by different frequency bands, whether they're depicted in absolute or relative or centroid terms. So each column here would depict one complex profile of one of several drugs. So the rows are here, the different columns are the different drugs. And you see that if you look at this, at the drug activity in this complex matter across bands and across different variables, you see very typical profiles where you have increases versus placebo in one cases and decreases versus placebo in others. And each of the drugs has the typical fingerprint-like pattern of where those increases and decreases are. And if you compare this now to a very similar picture depicting now the changes, the differences of brain activity in terms of EEG between groups that exhibit a certain mental disorder and controls. So you would see that some mental disorders, so each column here would now represent one of those mental disorders. Some of them exhibit certain increases in certain bands as compared to controls, and decreases in other frequencies bands as compared to controls. So again, you see a typical pattern characterizing each disorder, which can then be matched with the patterns that the drugs show vis-a-vis placebo. And here's a more concrete example. Here you have generalized anxiety disorder versus controls. In this case, you see the typical increased pattern in the alpha band and the total power, and you have the profile of somnium, a certain drug class, versus placebo, and you see wherever there are increases here in the disorder, the drug counteracts with decreases in the respective bands. So this illustrates this very interesting key lock principle. For every lock, meaning disorder, to be treated, there's likely to be a key, meaning a certain drug class that is, according to this profile and according to this fit, likely to be helpful or to be treating this disorder in the right and proper way. So this takes all these questions together of where and how in a very concise manner, and illustrates the power of the complex patterns of endpoint variables that EEG can have. Now, we may ask, where is the best place to implement an EEG measurement? I hinted upon before already that a natural place for an EEG in a drug trial would be an early phase 1 trial in a phase 1 unit, first in man or dose-finding studies. But that's not the only place. On one hand, I'd like to point out that EEG is also widely used in the preclinical arena. So you can measure rodent EEG or even primate EEG. You can measure rodent sleep and create certain translational hypotheses, deducing from what you see in a rat, for instance, to what you're likely to see in the human. So EEG is already a valuable tool in the clinical phase, helping you to lead you in a very efficient manner into your phase 1 studies. But also beyond phase 1, phase 2 can also be the place for EEG measurements when it comes to dose efficacy relations, dose finding, the therapeutic outcome, or the relationship to other clinical variables. So this wide portfolio of phases is typically where you would expect to find EEG as a measurement in your drug trial. Now, the final point I would like to make is that, of course, EEG, as you can see, since it can answer very many interesting questions, it is a very sensitive instrument for being able to do that, sometimes even too sensitive, in a sense that it can pick up other things as well. And therefore, it's of utmost importance in your trial that the data quality is high quality, is high, and is constant. So when applying EEG, as simple as it is in terms of costs and procedures, nevertheless, you need to look after all the procedures done in a proper way, of how the electrodes are patched, of what devices you're using, whether they also have the right kind of settings, from the impedance to filter settings, that you avoid artifacts that would distort your signals, that you keep subjects awake when your goal is to pick up awake EEG. It's not always the case. I mean, you can perform EEG equally in sleep. That's a topic that we're also very proficient in, but not the subject of this webinar here today. But the typical Pharmaco-EEG subject should be awake, and so we have to take care that they are, and that there's no other type of disturbance. So here's an example, for instance, of artifacts, a simple artifact like eye movement artifact, simple blinks, as you could see them in an EOG, in the measurement of eye movement directly. You see how it clearly affects most of the frontal polar and frontal channels, and thus distorts the spectrum and would distort your main endpoints that you want to calculate. So ideally, you would like to remove those artifacts and just analyze the EEG, the pure cerebral activity, free from such artifacts. It's just an example with respect to how important data quality is. And this is exactly now where we come in, why we offer services in this area, we being the Siesta Group, as a company, as a clinical trial service provider specialized in sleep and EEG. So these two things are exactly what we do on a daily basis, and we help you with services to implement EEG as an endpoint measurement in the clinical trial. So we offer a range of services. In particular, we can rent out devices if needed, if you're working with a site that doesn't have the devices yet or not for the duration of the trial. We can train personnel to do the right kind of quality control, and we do the centralized data analysis, and we do data quality monitoring, ensuring the constant quality, constantly being in feedback with the sites, potentially retraining them, and so ensuring the utmost information to get out of your whole procedure. Our strengths are that the people in our team have been doing this, some of them for over 30 years, in particular our Chief Scientific Officer, Dr. Peter Undera. We're using state-of-the-art technology. We also have software developers on board that stay on top of the development when it comes to computer-based analysis, like the spectral analysis that is paramount here. Here's a depiction of the typical data flow. So we, Siesta, we would be in direct contact with the investigator site be it in a single site or in a multi-site trial. We would receive the data via secure internet-based interfaces. We would feed back to the site whenever there is a certain endpoint that they need to know in order to continue the trial, to make, for instance, an inclusion decision or a dosing decision. But most of the endpoint variables would be calculated offline and then delivered directly to data management of the sponsor. So these many variables, as you've seen them, and they can be easily up to the dozens or even hundreds, they would not go the way via the CRF to the database, but directly, and we would agree with the sponsors on a previous, at this onset of the trial, on the exact set of variables that one would be interested in to look at finally. I should also mention we are not a CRO ourselves. We are also not a site. Also, we work with a few of them, and we can also recommend sites. And in particular, if anybody's interested in carrying out a trial in the U.S., we have a partnership with a very focused CRO called Community Research in the Cincinnati, Ohio area. They have a year-long experience in sleep and EEG, have a 60-bed phase 1 unit, and we work seamlessly with them, supplying them with the necessary know-how in the EEG arena and do the data processing. So if the full package is required, including a site in the U.S., we can offer something together with community research. Your advantage of working with us is our expertise, our utmost quality through our joint SOPs with working together with community research. The cost would be reduced since we are set up to work with that phase 1 unit already, and the study can start much more quickly. And we have a combined experience in almost every field of CNS-active drugs. Again, referring to us, Siesta Group, together with community research. So this brings me to the end of this presentation. Before I ask if there are any more questions, I would like to state our offer. We say also on our webpage, till the end of March, it's still a time that we name the EEG Awareness Weeks. So this webinar is part of our activity as part of those weeks. And we offer a free feasibility analysis for whoever is interested in looking whether and how EEG could or would fit into one of their planned trials. So if you provide us with a protocol, could be real or a mock-up. If you don't want to share at this point of time, of course we can sign confidentiality agreements and would have no problems in looking at real protocols, but sometimes a protocol outline, which is still a mock-up, is also sufficient. And we provide basically a general comment about the feasibility and suitability of EEG in its particular study, and we would calculate a cost estimate to show you how cost efficient such a measure could be carried out in this particular trial. Here are our contact details, both in Europe, where we're based, but also in the U.S., where we work not only with community research, but operationally in our services, also with Philips Respironics in Oregon. And would now like to ask you, thank you very much for your attention, and ask if there are any questions from the audience. For that purpose, I ask you to unmute yourself and just raise your question. I'd be happy to discuss. On the other hand, also, we will leave this presentation. We have recorded it. We will leave it on the web for you to revisit at any time, and we will also make the presentation available in PDF, downloadable at our website. So any questions that arise later, we will be happy to answer offline, of course, by contact or email or phone.